Hello, my name is Michael Downey and it's a great pleasure to welcome you all to the 11th AWRI webinar for the 2018-19 program. This session will take a look at grape-derived glycosides as a potential new source of flavour in white wines. Now, before I jump over to our speaker for today, some very quick reminders for the audience. To provide a comment or to ask a question, please open the Q&A part of the toolbar. Type in your question and click to send it through. If you'd like to get involved via Twitter, please use the Twitter handle @the_AWRI. The webinar is being recorded and will be available to view later this afternoon from the AWRI's YouTube channel. All registrants will receive a link to view this recording. I'd also like to thank and acknowledge Wine Australia for providing funding and support for AWRI webinars. Okay, for those that have just joined us, welcome. The title for today's presentation is Increasing Wine Flavour with Glycoside Additions. So for the first time, glycosides were extracted from Gewürztraminer, marc, and added to white juices prior to fermentation. And it's fantastic to have Mango Parker here with us today to discuss results from those trials. Mango is a senior scientist here at the AWRI and has been involved in a variety of research areas, including tannin reduction, white wine phenolics, smoke taint, and some others. So enough from me, I'll hand over to Mango to get us started.
Welcome to this webinar from the Australian Wine Research Institute. I'm going to talk about glycosides, wine flavour, and grape marc. I've divided the talk into two sections. I'll start by introducing glycosides and explain why they're interesting and important to understand in the context of grape and wine flavour. I'll describe a series of experiments that demonstrate how glycosides can be important to flavour during tasting. Then in the second half, I'll shift focus to practical application of this knowledge and describe how we used glycosides from grape marc to boost flavour in wine we made in a small-scale winemaking experiment. Flavour glycosides are non-volatile flavour precursors. That is, they have no aroma themselves, but can produce aroma under certain conditions. Glycosides are too heavy to be volatile, so can't be detected using the sense of smell. They consist of two parts, a volatile aroma or odour molecule, and a heavy sugar molecule that are chemically bound together. It can be useful to think of the glycosides as aroma compounds chained to a heavy ball. Glycosides are formed in the grape, are transferred into the juice when grapes are crushed, and persist in the wine. If the glycoside chemical bond is broken, the aroma chemical and the sugar molecule are released. This can happen during winemaking through the action of enzymes and acid, which unlocks the aroma from the grapes, giving the wine recognizable varietal aroma. Commercial winemaking enzymes are available that aim to break down glycosides and release maximum aroma from the grapes. Glycosides can also break down to release flavour in the mouth. Enzymes in saliva can break glycosides apart, releasing sugar and aroma molecules. The aroma molecules in the air in the mouth move to the nose via the retronasal passage. And flavour is experienced when the aroma is detected by the sense of smell, the olfactory system. When we are tasting a wine, we pay attention to the smell and the taste when we take our first sip, and we might enjoy the flavours for up to, shall we say, 2 minutes afterwards. So if glycosides are going to give us a flavour experience when they break down in the mouth, it needs to happen within 2 minutes of tasting the wine. We might refer to the flavours that develop in the mouth as the aftertaste, although strictly speaking, this is not using the sense of taste and could be more accurately called after odour. We have shown that glycosides can indeed break down in the mouth, releasing flavour. I think it's fascinating, and I'd like to explain some of the experiments that have shown this. We decided to concentrate on the aromatic variety Gewürztraminer, which is known to be high in glycosides, especially monoterpene glycosides. Monoterpenes are important aroma compounds that have fruity and floral aromas. We extracted glycosides from Gewürztraminer juice and wine. Then we purified the glycosides to remove any residual aroma that might confuse our experiments, and we removed bitter phenolic compounds. Then we measured the glycosides in the laboratory to make sure they were there. We checked that the glycosides could release volatile aroma compounds when we incubated with enzymes and saliva. The monoterpenes geraniol, nerol, and alpha-terpineol were among the aroma compounds released. So we knew we had glycosides capable of releasing flavour. The next step was to test the flavour effect using a sensory panel. We wondered whether the glycosides could break down quickly enough to release a noticeable flavour in the first 2 minutes after tasting the sample. To find out how the flavour from glycosides changed over time, we chose a sensory method called time intensity. The sensory panel was asked to rate the intensity of overall fruit flavour continuously over 2 minutes. After numerous training and practice sessions, the panel was presented with the glycosides to taste. We used the glycosides isolated from Gewürztraminer wine and juice at high concentrations, approximately 5 times the original concentrations. We also included a pure sample of geranyl glucoside, a pure compound, which can release the fruity floral monoterpene geraniol, again at a high concentration. We wanted the flavour to be obvious in our first sensory tests. We dissolved the glycosides in model wine with 10% alcohol and a pH of 3.5. We also included a model wine without glycosides as a blank in the study. The panelists were presented with 1 sample at a time with appropriate weights between samples. The samples were assessed in triplicate in a randomised order. Looking at the results, here is a summary of the panel mean data. In other words, the ratings from all the panelists averaged for each sample. Overall fruit intensity is on the y-axis, plotted against time on the x-axis. Reading the graph from left to right, we can see how the intensity of fruit flavour changes over time. A similar pattern is seen for the 3 samples. The fruit intensity starts close to 0. Then at between 5 and 12 seconds, the fruit intensity begins to increase, reaching a maximum at around 10 to 30 seconds, plateauing, then decreasing until the intensity reaches 0 at 50 to 80 seconds. The model wine blank shows a low level of noise, but confirms the effect is not due to the model wine matrix. The results were statistically significant and confirmed that glycosides were broken down in the mouth, releasing a burst of flavour within 2 minutes. When we looked closely at the raw data from each panellist, we could see that 6 of the 11 panellists were reproducibly experiencing this flavour effect, but 5 of the panellists were not. We repeated the time intensity sensory experiment again, this time using Riesling and Gewürztraminer glycosides at concentrations found in wine, and in combination with wine aroma volatiles. A different panel of 12 people was recruited and trained. This time, the panel mean results of the glycosides were not statistically significantly different to the model wine blank. But not all judges were rating the samples the same. Close inspection of the individual judge responses showed that 5 out of 12 judges were reproducibly experiencing flavour from the glycosides. This is an example of one judge who found the combination of glycosides plus volatiles to have the greatest intensity of fruity floral flavour, greater than the flavour from the volatiles alone or glycosides alone. Could it be that only half the population can taste glycosides? Perhaps some people are unable to break down the glycosides in their mouth, or don't transfer the volatile via the retronasal passage. Or there is some other factor stopping them from detecting the free volatiles, like the time frame. Or perhaps the non-tasters simply are not sensitive to the free aroma molecule. Large inter-individual variation has been observed for salivary flow, saliva buffering capacity, oral bacterial populations, swallowing and breathing behaviour, which could all influence the flavour release from glycosides. As a bit of an aside here, I know there are a number of winemakers tuned in today, and I'm interested in knowing if any of you have a special technique for retronasal flavour. Some winemakers and beer tasters have developed special ways to transfer more of the air in the mouth to the nose. I'm happy to go into more detail on how to train yourself to do this if anyone's interested. Coming back to the variation in response to glycosides, we decided to investigate this by testing a larger group of 39 people using 3 types of glycosides. We chose the fruity floral Gewürztraminer glycosides, fruity floral geranyl glucoside, and the smoky guaiacol glucoside. The glycosides were presented at reasonably high concentrations again, but this time in water. The glycosides were tasted individually in triplicate over several days. The panelists rated the terms fruity floral and smoky. For each judge, the ratings of the samples were compared to the control using analysis of variance with p less than 0.15 or significance of 85%. 54% of judges responded to the Gewürztraminer glycosides. 46% of the judges responded to the geranyl glucoside, and 64% of judges responded to the guaiacol glucoside. But it wasn't the same people who responded to all of the glycosides. When we put the pattern of responses together, we got a complicated picture. 28% of the judges responded to all 3 glycosides. 23% of the judges didn't have a significant response to any of the glycosides, although they may have rated flavour on 1 or 2 occasions. The other 49% responded to a combination of 1 or 2 of the glycosides. We found this very interesting and are currently working on experiments to help us understand these observations. So to summarise, glycosides are non-volatile flavour precursors with no smell of their own. They come from the grapes and are transferred to the wine. When the chemical bond between the sugar and the aroma molecule is broken, the aroma molecule is released, which can be detected by the sense of smell. We have observed inter-individual variation in sensory response to 3 glycosides, with 77% of people responding to at least 1 of the 3 glycosides tested. Are there any questions before we move on to the second part of the talk?
Okay, thanks, Mango. As Mango's indicated, we are going to take a very brief break here just to give opportunity for anyone that's got a question regarding the first part of Mango's presentation to ask a question. So if you do have any questions at this stage, please send them through. A quick reminder to ask a question, just open the Q&A part of your webinar toolbar, type in your question and click the send it through. I've got a question here, Mango. You spoke briefly about monoterpene flavours. Are there any other wine flavours that come from glycosides?
There are lots of different wine flavours that come from glycosides. It really depends on the variety. So the monoterpenes are the compounds that give that characteristic muscat Gewürz character. Other glycosides other compounds that come from glycosides include the famous aged Riesling character, TDN, also norisoprenoids, and there's a whole host of others. So yeah, it really depends on the variety, but there are many other different wine aromas that come from glycosides.
Okay, great. Thanks for that. I've also got a question here asking whether any correlation with age and gender.
I don't think so. No, there was nothing obvious about the group that we could use to explain that pattern of response.
Okay. You also spoke briefly about techniques for training yourself to better experience retronasal flavour. Do you have any, can you provide any quick insights around that for the audience?
Okay, so if you're curious to know exactly where your retronasal passage is, can I suggest that you taste some wasabi? I'm sure everybody who's tasted wasabi knows the feeling. It's quite unlike anything else. And what's happening when you taste wasabi is you are activating the trigeminal nerves along your retronasal passage. So that's a good way to feel where that is. And another thing that you can do is try and be aware of what you do with your breathing when you're tasting. Try and be aware of moving the air from your mouth up through your retronasal passage so it goes to your nose. Something that can help would be if you know what it feels like when you close your nose off to swim underwater. That's the feeling of closing the soft palate. So if you can practice doing that and then opening it, that can also help you to be aware of how to maximize that.
Okay, great. Some really helpful advice there. We might move on from here. So back to you, Manga, and we'll proceed with the second part of your presentation.
Okay, let's move on to the second half of the talk where I'll shift focus to a more practical application, a research experiment where we extracted glycosides from grape marc and added them to wine. Grape marc consists of grape skins, seeds and stalks that are left over once the juice has been extracted for winemaking. Thanks to Wine Australia's 2018 vintage report, we know that in 2018, Australia's wine grape harvest was 1.8 million tonnes, which means around 600,000 tonnes of grape marc were produced. Grape marc is usually used for low-value products such as animal feed, compost, or recovery of tartaric acid or ethanol. Grape marc of the variety Gewürztraminer has high concentrations of glycosides, but it also contains undesirable phenolics. In our winemaking experiment, we extracted glycosides from Gewürztraminer grape marc, removed phenolic compounds and aroma volatiles. We added the glycosides to 20-litre duplicate ferments of Riesling juice. We added the glycosides at 2 time points, to the juice prior to fermentation and to the wine at bottling. We thought that glycosides added to the juice would have a greater chance to break down and release aroma during fermentation, and that the glycosides added at bottling would break down less and produce wines with lower fruity or floral aroma and more flavour in the mouth or aftertaste. We also included 2 even more extreme treatments. One was a double addition of glycosides to the juice, and a treatment where we added a single dose of glycosides to the juice and a single dose to the wine. Glycosidase enzymes were not used in the winemaking. Thanks to WIC Winemaking Services for doing such a good job of making these wines. All the wines fermented to dryness and the glycosides did not change the fermentation kinetics or the basic wine chemistry. We analysed the wine 3 months after bottling to see what effect the glycoside additions had on the glycoside concentration. I'll concentrate on geranyl glucoside because it was the main glycoside in the grape marc extract. The geranyl glucoside was quite low in the wine before we added the glycosides, and the increase was directly proportional to the glycoside addition. The timing of addition was not important, showing that most of the glycosides were not broken down during the fermentation. That was surprising because we expected greater glycoside breakdown during fermentation. When we look at the sensory rating of floral aftertaste, we can see that this follows the concentration of the glycosides. Looking at the free volatiles, we expected that some breakdown of glycosides would happen due to the acidic conditions in the wine, so we should see an increase in the free aroma molecules due to the glycoside additions. We also know that a number of monoterpenes can form in wine because the acid facilitates chemical rearrangements. I've shown linalool as an example of the monoterpenes. The control wine had a concentration of approximately 20 micrograms per litre of linalool, and each single dose of glycosides increased the concentration by about 50 micrograms per litre. This is reflected in the floral aroma sensory ratings, although the biggest change came from the single addition compared to the control. We checked this panel to see who could perceive flavour from the glycosides and divided the panel into 2 groups: tasters of geranyl glucoside and non-tasters. Bear in mind that there are only 5 tasters and 6 non-tasters, and there were some differences in how the 2 groups use the sensory scale. By tasters, I mean those who can perceive flavour from the glycosides using olfaction, the sense of smell. I'm not referring to the sense of taste here. We can see that the glycoside additions have increased the floral aftertaste much more for the tasters than the non-tasters. To summarise, glycosides increased fruity, floral aroma and aftertaste, especially for those able to taste glycosides. The timing of the addition had a relatively small effect. There was no significant increase in bitterness and astringency in the wines. And this opens up the possibility of utilising grape marc as a source of flavour precursors. If you'd like to find out more about this work, it has been published in the following papers that can be accessed for free via the AWRI library. You'll see the AWRI library email address for those who'd like to access this fantastic service. I'd like to thank the many people who've contributed to this work, in particular my PhD supervisors, Miguel de Barros Lopez from the University of South Australia, Lee Francis, Josh Hickson, Corey Black and Marcus Herderich from the Australian Wine Research Institute. Thanks to the sensory panellists and AWRI colleagues. This work is supported by Australia's grape growers and winemakers through their investment body Wine Australia, with matching funds from the Australian Government. The AWRI is a member of the Wine Innovation Cluster in Adelaide. This work was also supported by the Australian Government Research Training Program Scholarship. I'd like to invite questions and comments. We're interested in hearing your opinions and suggestions and always welcome collaboration with our research, either now or outside of this session. My email address is displayed and I'm happy to answer questions via email.
Okay, fantastic. Thank you, Mango, for walking us through the second part of your presentation there. We're going to move from here into another Q&A, so Mango will stick around for a little while. If you've got any questions, please start to send them through. We've got a couple here already. Mango, one would like to see the references again, so perhaps you could slide back to that previous slide what I might do is actually include these references in the follow-up email. So you'll have those to view then. And if you'd like to request any of those, then yeah, please do contact the AWRI library. Okay, so we've got a quick question here, Mango, about how the glycosides were extracted. Can you provide any comments around that process?
It was a fairly simple procedure, and we did it in the winery that we have just up the road here, the Wick Winemaking Services. So basically we used water, ethanol, caustic, and a resin for the procedure. If you'd like to know some more details, yeah, please talk to me directly and I'll be happy to explain what we did.
Okay, fantastic. Thank you, Mango. Yeah, as Mango's indicated, please do contact her if you have any questions that you think of after the session, then yeah, please do contact Mango directly, and I will include her contact details in that follow-up email as well. We don't have any other questions, but we'll stay on the line for another 30 seconds or so. So now's your chance. If you've got any questions, please send them through. Okay. So we do have another one here, Mango. What about using glycosides in non-Gewürztraminer white wines?
Well, in our experiment, we added it to Riesling, and actually we tried it with a Chardonnay as well. And the Gewürztraminer character from the glycosides was very obvious. So there's a number of things to discuss there about whether you would want to do that. It would also be really interesting to look at different varieties. So we're doing some research into that at the moment, looking at the glycosides that are available in marc of different varieties and which ones would be interesting and useful to extract.
Okay, great. So perhaps stay tuned for some more on that. Okay, another question here, Mango. You mentioned that the glycoside concentration results were directly proportional to the addition rates with no breakdown, but was there any increase of linalool concentration? How can that be explained?
Okay, yes, there must have been some breakdown of glycosides in order to release the free volatiles that we saw. But the concentration of glycosides that we dosed was very high. Perhaps I'll just go back to that slide. Excuse me. So a single addition resulted in a concentration of geranyl glucoside of around about 2,000 micrograms per litre. And the increase that we saw in linalool was around 50 micrograms per litre. So it's such a small amount proportional to the total that was there that really it was insignificant compared to what remained. I hope that makes sense.
Yeah, I think so. But Joel, if you want to follow up with Mango directly, if anything wasn't quite clear there, or you'd like to get some more information around that, please contact Mango directly. Another question here around whether skin contact during fermentation naturally extracts glycosides without the addition of ethanol and resins.
Absolutely, and yes, that's one technique that can definitely be used. It's probably being used straight away to increase more flavour. The glycosides will readily transfer into the juice from the skins. the problem with doing a lot of skin contact is that you may get undesirable mouthfeel attributes from other compounds like phenolics in the grape marc. The amount of glycosides that we added would be equivalent to a 24-hour full skin contact maceration pre-fermentation. So for a variety like Gewürztraminer that's quite phenolic, that would have a significant effect on the palate. You'd be looking at significant bitterness from that. We wanted to try and isolate the glycoside effect from any of the other mouthfeel effects in our experiment. So we went to the effort of removing the phenolics, but that's definitely something that could be explored more. And another option would be to do more skin contact and then look at finding options to try and remove the phenolics later.
Thanks for that, Mango. Do you have any information, or can you share where this project might be heading next? Are there any industry trials or anything that's on the horizon that you can share with us?
Yes, so we're looking at different varieties. So we want to understand which varieties would be interesting and worth pursuing. And we also want to understand more about the stability and the behaviour of the glycosides over time. So if people were to add them, what would the wine and the glycosides look like 3 months, 6 months, 1 year down the track? So looking at the stability there, those are the kinds of things that we've got planned. Also, we've got a big experiment which we're finishing up that's trying to understand more about the variation in the sensory response to the glycosides. So that's happening as well. That's going to be really interesting to see, looking at thresholds, sensory thresholds, and the microbial population of people's mouths. Stay tuned.
Fantastic. So we'll look forward to some further results from this work. I think we'll probably leave it there, Mango. We've exhausted all questions. So if you could flip forward to that final slide there for me, that would be great. I'd like to first extend a big thank you to Mango for sharing results from these exciting trials. And I'd also like to thank you, the audience, for participating in today's session. Attendees will receive a follow-up email with a link to this recording. The next AWRI webinar is on Thursday, 6th of December, with the Bureau of Meteorology's Darren Ray providing a seasonal outlook for vintage 2019. Now, this was a very popular session when we ran it last year, so if you'd like to register, please visit the AWRI website. Thank you again for participating, and I look forward to seeing you at the next AWRI webinar.